broadcast has started. We are now live. Oh, good to know. All right, so I'm assuming everybody can see and hear me now. My name is Greg Cox, and I wanted to thank Morit for the introduction earlier. We were trying to get ourselves all connected this morning, and so I think we're all here and ready to go. I'm incredibly excited to have Dr. Sheena Chu here with us this morning, and she's an ALS physician and researcher at the Sean M. Healy AMG Center for ALS at Mass General Hospital. She's board certified in neurology and in neuromuscular medicine, most appropriate for today's talk. Dr. Chu received her medical degree at Harvard Medical School, and she completed a residency at Brigham and Women's Hospital, followed by a fellowship at Brigham and Women's Hospital as well. Her ALS clinical research fellowship is at Mass General Hospital. She's currently working at Mass General, and we're really excited to hear about her own clinical experience, as well as the latest data that's coming out in ALS research and the clinical trials that she's participating with and guiding, and the platform trials. So we're incredibly excited to hear from Dr. Chu this morning, and I will let you hear from her because she's why you came here this morning. So I will leave and let her tell you all about the research going on. Thank you.
Thanks so much, Dr. Cox. Hi, everyone. I'm so excited to be here with you all. So as he said, I'm Sheena Chu. I'm a physician at Mass General in the ALS Center. And I spent all of my time taking care of ALS patients and doing research to advance drug development for ALS. So I'm so happy to be here with you today. On a personal note, I became involved in ALS because about 2 decades ago when I was in college, I had a good friend who was diagnosed with the disease. And things are really different now than they were back when my friend was diagnosed. There are new developments in ALS clinical trials that make me feel hopeful in a way now that I didn't feel when he was diagnosed back then. So I'd like to share these with you today. Just a quick disclosure to start, I get research funding from the American Academy of Neurology and Biogen. So historically, ALS therapy development has been very, very challenging. When my friend was diagnosed in the early 2000s, there hadn't been a new drug approved for ALS since 1993, and that was the only one that existed. And from that time, for the next decade, there were dozens and dozens of trials for ALS that weren't successful until just 3 years ago when edaravone was approved. And that was very frustrating, to say the least. And it still is now. I'd like to share, though, how this landscape is changing and some of the advances that have led the way. So just an outline. First, I'll briefly touch on advances in science that have changed the way that we think about developing drugs for ALS. Then I'd like to spend the majority of my time with you telling you stories about 3 ALS trials that I think are changing the way we think about ALS research and giving me some hope, namely the Biogen tofersen trial, phase 1/2 study results for genetic ALS SOD1 mutations. I'd like to tell you about the Amylyx AMX0035 study results and some new data that just came out today. And I'd like to tell you about the Healy ALS platform trial that was just launched this summer. And then I'd like to leave some time for questions. That's my favorite part. So let's start with some very brief advances in the science. One thing that's changed in the last decade is that we've been getting a better understanding of the basic biology of ALS. And one way that we're doing that is discovering more genes that cause the disease. So back in the 90s, when the first gene was discovered, the SOD1 gene, we only knew that one mutation that caused ALS, and that causes ALS in about 2% of people overall. So we didn't know that much about the genetic causes of ALS. In the last decade, that the rate at which we're discovering new genes has really been increasing because the science is better. And I think we have more than 18, maybe more than 20 genes that we know are associated with ALS now. It's important to note that even though we know a ton of genes associated with ALS, only 1 in 10 people with ALS have a gene mutation that we can identify. So in reality, most people with ALS don't have a gene associated with it. But still, even if most people don't have a gene associated with ALS right now, knowing about the genes that cause disease in only 10% of people helps us understand the disease as a whole and helps us make better drugs for everybody. And this is because when we identify a gene that causes ALS, when it's mutated, meaning when a gene isn't working normally, then we can ask, what does that gene normally do in the body? So these are the genes that, some of the genes that we've found to be associated with ALS. And then we can say, all right, what does that gene normally do? What's its normal function? And then using this approach, we've learned that the normal functions of the genes are involved in lots of the processes involved in this picture, like energy metabolism in the cell, processing of proteins and something called RNA in the cell, transporting proteins up and down the cells, and then inflammatory responses to injuries in the cells. These are all things that we've discovered through genetic abnormalities. So we think these are all related to the disease process in people with ALS. Then we can ask, well, can we develop a drug that protects that function for all people with ALS, not just the people with the gene mutation? So because we know more about the science, and also because there's been huge engagement with the community, the ALS community, and a big injection of funding into the research after the Ice Bucket Challenge in 2013, 2014, ALS research and drug development has really been gaining momentum. There are more than twice as many companies designing drugs for ALS now than there were even just 5 years ago. So it went from 60 to more than 120 now. So there's lots of activity, lots of interest finally, and some of these treatments are finally coming into clinical trials and being tested in people. And so that's really been a huge change from the time that I was first exposed to ALS and now. So there are many different clinical studies ongoing because of this momentum, and I'll just highlight 3. Each of the clinical trials I'm going to talk about represents a new direction and a new strategy in therapy development for ALS. For example, the Biogen tofersen study is targeting a specific gene mutation, and this is a phase 1/2. I'll tell you about that in a second. The Amylyx Centaur study of AMX0035 for ALS mobilized a whole community to get that study up and running. And the Healy ALS platform trial is an innovative trial design that we're using to accelerate how quickly we can test drugs in people with ALS. And I'll tell you about that last. I'm excited about all 3 of these. Before we start talking about all of the studies in general, I wanted to first define the process for getting a drug from animal testing all the way to FDA approval. So drugs classically go through several stages of testing in people. Phase 1 is the first thing that we have to do to get a drug from animals to people. It's usually tested in a small number of people to check and see if this drug is safe. And then also that phase 1 study is done in a small number of people to get a sense of what's the right dose for people. So you usually start with a low dose and go up and then test to see what the drug levels in the blood and the body are and make sure that it's safe in people. Once it passes phase 1, then it usually is tested in phase 2 in a medium-sized group of people to further test whether it's safe in a larger group of people, and then maybe get a sense of whether a drug might work for the disease. And then phase 3 is the big trial, the classical ones you hear about in the news, where you test a drug in a large group of people, and the question you're trying to answer is whether the drug works, and then also to keep making sure that the drug is safe. Classically, the FDA requires 2 separate phase 3 studies in order to get a drug approved. So it's a lot of people and a lot of time, and there's a lot of work in the ALS community to see if we can sort of bypass the classical rules, and because there's so much need in ALS, and that's a completely separate discussion that I'm happy to answer questions about afterwards. But this is the classical stages of testing for most drugs before they can get approved and sent to people. Okay, so let's start talking about the trials. I'm going to start with the Biogen tofersen trial for patients with SOD1 mutation. So this is phase 1/2, meaning these are early. This is super early in the development process where we're testing safety and maybe getting a hint of seeing whether it works. This study was just published this summer in the New England Journal of Medicine. And I'll tell you a little bit about the drug. Tofersen is what's called an antisense oligonucleotide, and this drug was designed to treat the rare genetic form of ALS caused by mutations in a gene called SOD1. About 2% or 1 in 50 people with ALS have mutations in this gene. So it's a pretty rare mutation. And we often think about this when ALS runs in families. So how does this drug work? Normally, your genes, which are encoded in your DNA, are transcribed into something called RNA, so say your gene, your SOD1 gene is in your DNA, then it's transcribed into something called RNA. And then the RNA is used as a template to make proteins. This is like a picture of a protein. So this would be your SOD1 protein that came from the SOD1 RNA, which was made from the SOD1 DNA. I'm going back to high school talking about this. So with the SOD1 mutation, there's an abnormality in the SOD1 gene in the DNA, which is then transcribed to the RNA. And then that leads to an abnormal SOD1 protein, which essentially we've discovered junks up the system. So in SOD1 ALS, the protein, which is abnormal, causes problems because it folds in inappropriately. So scientists have realized it's actually the presence of abnormal SOD1 protein that causes ALS in this group of people with SOD1 mutations. So they designed this drug called an antisense oligonucleotide called tofersen. So tofersen is injected into the spinal fluid by spinal tap, by lumbar puncture. And the goal is that it goes in from the spinal fluid into the neurons of the brain and spinal cord, and then the tofersen binds to the abnormal RNA. And what it does is it causes the RNA to be degraded. And that way, the abnormal, the abnormal SOD1 protein can't be produced. So the educated guess, the hypothesis, is that injecting tofersen into the spinal fluid of people with SOD1 ALS will reduce the amount of this abnormal SOD1 protein. And the hope is that this will get to the root cause of the disease in people and treat the disease. So that's the guess, and that's the hypothesis we're testing with this trial. So how did the scientists and the doctors test this hypothesis? The study design was a randomized placebo-controlled trial. Placebo means sugar pill, or a substance that looks like the drug but doesn't have a therapeutic effect. And trials use placebos to compare what happens to people who get the drug versus people who didn't get the drug. So in this study, people who had confirmed SOD1 mutations that caused ALS were screened for the study. If they qualified for the study, then they were randomly assigned 3 to 1 to tofersen or placebo. And over 3 months, these people got 5 injections of either tofersen or placebo. They didn't know, and the doctors didn't know what they got either. 5 times over 3 months. And because this is a phase 1-2 trial, they were trying to figure out what dose is correct. So participants received different doses. Some participants got 20 milligrams of tofersen, some got 40, some got 60, some got 100 milligrams of tofersen, and they started with a low dose and went up to make sure that it was safe even as they went up on the dose. After these 3 months of injections, then everybody was followed up for 3 months. So what did this study look for? This is, as you remember, an early phase study. So they were looking at first and foremost, safety. Then they also wanted to look and see, how much drug got into the body. Did the drug affect proteins in the body? And then as an exploratory thing, they were just trying to get their feelers out. Does this drug actually affect people's lives? Does it affect their physical functioning? How did they do that? So for safety, they looked at adverse events reported by patients, by lab tests and other assessments. The results were that, the big picture is that near every participant in the study had some sort of adverse event. And most commonly, these were things associated with the lumbar puncture, the spinal tap procedure, like pain and headaches. The adverse events that were more common in the tofersen group compared to the placebo was increased white blood cells, so a little bit of inflammation in the spinal fluid compared to the placebo group. So this is being looked into further with further studies. But overall, the drug was relatively safe and well tolerated, and most of the issues were with the procedure itself. The study then looked at what the drug did in the body. So they looked at levels of the drug in the blood and the spinal fluid. And then they also looked to see whether the drug had an effect on the protein in the body. If you remember, the hypothesis was that if you injected the tofersen into the spinal fluid, it would get into the cells and stop the abnormal SOD1 protein from being made. So they wanted to see whether SOD1 protein levels changed in the spinal fluid. So here's what they learned. They learned that larger doses of tofersen had larger decreases in SOD1. So this is good. This is what they were hoping for. And this is what we call a dose-dependent effect. It tells us that the drug is doing what we think it would do, which is decrease the amount of abnormal SOD1 protein. So this is the placebo group is in blue. It's a little bit hard to see here. And then 20 milligrams is in green, 40 milligrams is in yellow, 60 milligrams is in black, and 100 milligrams is in orange. And it's not perfect one-to-one, but the higher doses of tofersen reduced the protein more. So we think that the drug is doing what it's supposed to do based on its mechanism. And finally, they did some exploring about whether the drug had an effect on people's physical functioning. So this wasn't the main goal of the study because it was a phase 1/2 looking at safety and the drug levels and drug effect on protein. But personally, I think this was the most exciting part of the study. So how did they look for it? They looked to see whether the drug had any effect on people's ALS-FRS-R score, which you might have done in clinic. The ALS Functional Rating Scale is a questionnaire with 12 questions asking how you do on daily activities like brushing your teeth, handwriting, walking, going upstairs, turning in bed, etc. And it's on a scale of 0 to 48. So they looked to see whether the drug had any effect on that score over time. They also looked to see if the drug had any effect on people's strength over time and breathing scores over time by measuring quantitative strength testing and vital capacity testing, the breathing test you might have done in clinic. So here are the results. This is kind of small. I took it from the paper. Looking at the rate of decline measured by that questionnaire, the ALS-FRS score over time, looking at the breathing scores here, and looking at the strength scores. So drug compared to placebo, placebo is in blue and tofersen is in orange. These graphs, which show just the people who received the highest dose, the 100 milligrams dose of tofersen, people seem to do better. Higher is better in these graphs. So they represent decline by ALS-FRS-R score, decline in the breathing score, and decline in strength scores. You can see that the decline is slower for people who were on the tofersen group compared to the placebo group. But what's really noticeable is that if the people in this group were divided into fast progressors and slow progressors, meaning the people whose disease was moving really fast versus people whose disease was relatively stable, the effect was really pronounced in the fast progressing group in this row. So the tofersen line is orange again, and the placebo line is blue. And this is a tiny group. There's only 4 people in the tofersen group here, and there's only 4 people in the placebo group. But even in just 4 people, you can see the difference between people who got drug versus people who got placebo. There was a big difference between the groups in ALS-FRS scores over time, breathing scores over time, and strength over time. The effect wasn't as pronounced in the non-fast progressing group in this third row because both groups were relatively stable. So this is exciting, but this effect was only documented in a very small number of people, 4 people, and this wasn't enough for us to get, for example, FDA approval. It's too small. So the next steps are underway. The phase 3 trial is actually enrolling right now. It's called the VALOR study. And they're testing tofersen, the 100 milligram dose in people with the SOD1 mutation. So if you know of anybody that has the SOD1 mutation, this might be an interesting study for them to be a part of. Once this trial is done, then they're going to try and see if they can get FDA approval. I don't know if they're going to do 1 study or 2, but that's the next step. Okay, so that was quite exciting. I think that's a big change compared to even just a year ago. But since the SOD1 tofersen study really only applies to 2% of people with ALS, just the people with the ALS SOD1 mutation, I'd like to move forward to a second study that applies to all people with ALS. This is the AMLX Centaur AMX 0035 study. It was just published a few months ago, which, cutting to the punchline, was a positive study for the whole ALS community. And breaking news, as of this morning, there was a new study that was published that showed that it actually increased survival too. So I just pulled a figure from the paper about 10 minutes ago, so I'll share that with you too. So this study was done across the United States through the NEALS Consortium, the Northeast ALS Consortium, coordinated by Mass General and Barrow Neurological in Arizona. And it was funded by the ALS Association, ALS Finding a Cure, and the company AMLX. what I love about this study is that it was a study that really only happened because the whole ALS community came together to see it through. It is just a quick note about the biology of this drug. It's a combination of two compounds, sodium phenylbutyrate, PB, and tar-ursodeoxyl, TURSO, which act by reducing something called endoplasmic reticulum stress and mitochondrial dysfunction. So we think that it has to do with cellular stress and energy metabolism. And the thought is that these two compounds in combination are better than each one alone. Just wanted to tell you the story of how this came to be. It's really an amazing story. You might have heard it. The company started out in a dorm room at Brown with two undergrads, Justin and Josh, who were the nicest guys, who were thinking about the science of neurodegeneration for class. And they came across these two compounds and did some research on it and thought, okay, these have a lot of promise for treating neurodegenerative disease, and maybe these two compounds are better in concert than each one alone. So they decided to create a company to test this idea. And they reached out to Rudy Tanzi, Dr. Tanzi at MGH, who is a leader in Alzheimer's research, who happened to be a fraternity brother. Apparently, that's the reason why Dr. Tanzi responded to them. And so they met with Dr. Tanzi, talked about the science, and then worked really hard to work on the science, partially in his lab, I think, or on their own, to prove that this was a good concept. Dr. Tanzi then introduced the two of them to Dr. Merit Sukovich, who's the chair at Mass General and the founder of the Healy Center for ALS. And Dr. Sukovich said, well, have you thought about using this for ALS? I've thought about these compounds in the past, and they seem really promising. So they did a lot of work. They hustled. They got funding from ALS Finding a Cure, Alzheimer's Cure Alzheimer's Fund, and then eventually got funding from ALS Association as well to do the animal studies. And then they got paired up with Dr. Paganoni and Dr. Sukovich at the Healy Center to design the trial, which started enrolling 2017, finished their last patient in December of last year, and then just recently published the results. And it was positive, which is amazing because there hasn't been a positive study except for the Radicava study 3 years ago. So the design was also a randomized placebo-controlled trial, but this was phase 2/3, so it's a little bit later in the process. So it was a bigger study because it was later. So they screened more than 170 people with ALS, and it was mostly more rapidly progressing people who met definite ALS criteria, which means they had weakness or symptoms in 3 parts of the body, usually the mouth, the arms, and the legs. Those who qualified were randomly assigned in a 2-to-1 ratio to the drug, which is AMX0035, or placebo, and they took the drug every day for 24 weeks. So it was a 6-month trial instead of a 3-month trial like the tofersen study was. So what did the study test? It tested the safety and tolerability of the drug, and they also studied the effectiveness, whether AMX0035 had an effect on people's physical function. So similar to the tofersen study, the AMYLIC study measured safety by adverse events reported by patients, lab tests, and other assessments, and then they measured physical function by the ALS Functional Rating Scale. The punchline is that treatment with the AMX0035, which is in red, slowed the rate of decline for people with ALS compared to people who are on placebo. So people on placebo had a decline of 1.66 points per month, and people on the AMYLIC drug had a decline of 1.24 points per month. And this is faster than the general public and also faster than previous trials because they enrolled fast progressors. So this was a significant effect. And in terms of safety, the most common side effects were related to this GI issues, gastrointestinal issues, nausea, diarrhea, stomach pain, and that mostly happened in the first 3 weeks. Overall, in terms of serious adverse events, people who received the AMX0035 experienced fewer serious adverse events than people receiving placebo. So overall, this drug was also relatively safe and well tolerated. And as a summary, people had a slower rate of decline, and then the side effects were primarily in the first 3 weeks and related to gastrointestinal issues. And this is the bit that I just learned about this morning. So this is called a survival curve. And essentially, they just published this morning that treatment, well, they followed people for up to 36 months after the study was over. They did something called an open label study where everybody who finished the trial had the opportunity to take the drug, and they followed people over a longer period of time, so up to 3 years from the time they enrolled. And if you look at the median survival, it looks like people who started on AMX 0035 had a looks like 6 or 7 month longer average median survival than people who were on placebo, and even the people on placebo who eventually received AMX 0035 during the open label extension. And this effect is on top of riluzole and edaravone, because nearly, I think 75 plus, maybe 80% of people in the study were already on riluzole and or radicava. So this drug, we think, is in addition to what already exists. So what are the next steps for this drug? I think that's the big question. And my understanding is that the company is in talks actively with the FDA to see whether or not they need to do another study, a phase 3 study, or whether this study by itself would be enough. And we can talk about that in the question and answer session as well. In our remaining time, I'd like to highlight something that we're doing at the Healy Center across the United States, and I'm really quite excited about it too. It's called the Healy ALS Platform Trial. This is something very close to my heart. We just launched this at Mass General and across the country this summer, and it's taken an entire community to launch it in the middle of a pandemic, with people desperately working really hard in the background to get it up and running despite all of the stuff that's going on in the world. This trial is a new kind of trial that hasn't been done before, this platform trial, and the goal is meant to speed up how quickly we can get to a cure. There are a huge number of partners that helped us get here, including the ALS Association, a number of companies, FDA. We've had a patient advocacy community. It's been really a community effort. The idea for a platform trial came from the fact that there are more than 100 companies, as talked about, working on ALS drugs. So we have a ton of drugs to test. There's a pipeline of drugs that we need to get to people. And that's where the bottleneck is. It takes a lot of work to get clinical trials up and running and a lot of time, and it's slow and inefficient. One thing we've done traditionally in ALS drug development is we have to set up the infrastructure for a clinical trial, then we test one drug, and then the trial ends, and then all the infrastructure goes away. Then the next drug to be tested needs to set up a whole new infrastructure all over again, setting up contracts with companies and sites, recruiting patients, getting approval from the hospital review boards before we can even start the testing. So it's all incredibly efficient with lots of lag time. Like if you were to build a stadium to play a single soccer match and then take the stadium down and build a new one before you play another match. So we thought, why don't we keep the clinical infrastructure and test multiple drugs at the same time, allow drugs to come in and out of this clinical trial, like building one stadium for an entire soccer tournament over many years, many soccer tournaments. This concept is called a platform trial. This has been done in cancer trials in the past, where you build one giant clinical trial and then allow different drugs to be tested using that same infrastructure. We designed this infrastructure to be as patient-friendly as possible and as efficient as possible. So we're starting with 3 different trials that are running at the same time in one big platform. And what happens for people who are interested in participating in the trial is that people are screened for the trial overall here. And then if they qualify for the trial, then they are randomly assigned into what we call a regimen, which is a standard trial of a drug versus placebo. So we have 3 regimens. They're called regimen A, B, and C. It's because there's 3 regimens currently running. If you were to qualify, you'd be randomly assigned to one of these 3 that are available. I think a 4th one is coming in a couple of months, so there would be 4 that you would be one of the 4 that you'd be randomly assigned to. So then you would say, okay, I've been assigned to regimen A. then the person would come back and see if they would be screened again for the regimen A and see whether they qualify for that particular trial. And if yes, then they'd be assigned in a 3-to-1 ratio. So 3 out of 4 chance of getting drug and 1 out of 4 chance of getting placebo in that regimen. And the regimen lasts 6 months, where you have very similar to the Amylyx trial, where you take the drug or placebo for 6 months in a 3-to-1 chance of getting drug or placebo. And then when the study is done, that 6 months is done, and then everybody has an opportunity to try that drug in an open fashion, so no chance of placebo after 6 months. What's cool for patients is that there's more access because there's more trials going on. This is going to be going on for a long time. And also, you have a higher chance of getting drug than placebo, 75% chance of getting drug, and then everyone who finishes the trial gets a chance to try the drug 100%. And what's cool for the scientists, the nerdy part, is that because all of the trials are going on using the same infrastructure, having the same kinds of visits, the same frequency of visits, the same assessments done at each visit, then the placebo patients from each arm can actually be shared. And that actually gives us more statistical power with fewer people getting placebo to answer the question. I'll tell you more about that in just a second. But a couple of other things about the platform trial is that there is a scientific advisory board that is reviewing all of the drugs that are applying to be in the trial. So these treatments are selected by ALS scientists and experts based on the best drugs that are available to be tested at this moment. The platform opened with 3 treatments, but the next treatment will be added soon. That'll be this fall. And then there are 3 more companies with drugs that are interested in joining the platform next year. So it's going to just keep growing, and we'll be testing more and more drugs until we find a cure. The overall goal of this trial is to work faster, to work on the ALS clock rather than the standard clock. We want to be as efficient as possible and to get to a cure faster. Our statisticians actually did a simulation to see how long it would take to get to test and find good treatments for ALS. So let's just assume that 1 out of every 10 drugs that we have to test for ALS is a good drug that slows disease progression by 30%. Using traditional clinical trials where you do one trial after another, and you need 120 people on drug, and then you'd have to have a matched 120 people on placebo, meaning you need 240 people. And it takes 3 months to take down and set up a new trial, one after another. To test those 10 drugs, it would take 12 years to get through those 10 drugs to discover the one good one. And it would take 2,400 people to participate in these trials, half of them on placebo. That's what we've been doing up until this point. Using this platform trial where we're constantly testing 3 at the same time, and each regimen, you get 120 people trying the drug and 40 people on placebo, because you're actually taking the other 80 people from the other 2 regimens and sharing them to compare against the 120. So 120 versus 40 from that regimen plus the 80 from the other 2 regimens, getting the same power that you would get from a traditional drug trial. It would actually just take 4 years to get through 10 treatments, and it would only take 1,600 people to participate. Out of those, only 400 would be on placebo. So it's just a faster and better way to get to an answer. It's a win-win for the participants who have more access to investigational treatments and a lower chance of receiving placebo than traditional drug development. And it's a win for science because we can test the treatments faster, we can get to a good treatment faster. So this is something new. We think platform trials can accelerate how quickly we can get to effective treatments for ALS. We've gotten a lot of support from all the players. The FDA has been very supportive of this approach. Our industry partners who have drugs to test, they're pretty excited about doing this too, to get an answer faster. Clinician scientists are really on board. There's a lot of excitement. And then the patient and family community. We've had an amazing patient advocacy board that has been helping us think through the design and make sure that it's humane and make sure that we keep the patients at the center of all of this. This is for you. and we're all working on this together. And this is a perpetual trial. So after the first 3 regimens are done, we'll have more added to it. And then as those are gone, we'll add more drugs to it, and we'll keep testing until we find a cure for all people with ALS. So it's a mammoth undertaking. It's just started, and we need people to partner with us to get this up and running everywhere. This trial is being conducted at 54 sites across the country, and more sites are being added. So it's not just happening at MGH. These sites are very experienced at conducting clinical trials for ALS. Right now, things were delayed because of the pandemic, unfortunately, but now 24 sites are open. If you're particularly interested, if you're interested in participating, this is a long link. I'm happy. Basically, if you Google Healy ALS platform and go to our website, you can actually find a list of all the sites that are open and are near you. The contact information for sites it'll be updated on our website as additional sites open. You can also find more information about the platform trial in the Northeast ALS website at neils.org under ALS trials. Or you can go to clinicaltrials.gov and then search for Healy ALS platform trial. And that also gives an updated list of all of the sites that are enrolling, plus contact information for those people at the sites. Because it can be kind of hard to find your way around, there's somebody, a patient navigator that you can contact at Healy ALS platform at Mass General Hospital, mgh.harvard.edu. And that person's job is to help find a site that is a good fit for you. And you can also sign up for our newsletter. It's the ALS Link newsletter. If you go to the Mass General, if you search Mass General ALS, there's something called an ALS Link, and we sent out periodic email newsletters. There's also webinars that happen each week, I think. The big picture is that we'd like to partner with you to find a cure for ALS. You're part of your team. We'd like to fight this with you. So I think that's the end of my talk. I hope that this was helpful for you. I'd like to make some time for questions if you have any. Thanks very much. And thanks to also for this opportunity to connect with you. Happy to answer questions now.
Thank you so much, Dr. Chu. And I know Greg is coming back on. I'm hoping everybody can see and hear us well. We do have, I think you answered this question of their current active trials for the MLX and where can they go. So you gave that information. If you've forgotten that information or need that information, your care service coordinators can give that to you as well, and show you how to sign up for the newsletter at MGH. We have a question from Roger. Any updates on Neurone trial at MGH?
So the Neurone trial has finished at MGH in terms of the patients who have been in the trial. Our site is wrapped up, but I don't know for the other sites across the country. I do know that I wasn't involved in the Neurone study, but I think what's happened is that they have to get the data cleaned and then analyze the data and then see whether that is good enough to take next steps. So at MGH, we're done seeing patients for the Neurone trial, and it's mostly data cleaning and all of the housekeeping activities after the trial is done.
Okay. We have a question from Bob Carty. Hi, Bob. Bob is also on our advisory council. So with all the ALS research being done on a solution for a treatment and vaccine for COVID-19, is there any data that we could get access to help reduce our timeline for an ALS solution?
That's a really great question. I think that one thing that has been very clear is that it's possible to accelerate things, because when we're dealing with the pandemic, all of a sudden all these accelerated pathways exist. And can we harness that kind of energy and that kind of precedent to do the same thing for ALS drugs? And I think that is on the minds of a lot of companies that are making drugs for ALS, and I think that's on the minds of advocacy communities too. My understanding is that that already the ALS Association is working with Amylyx to see if there's an accelerated approval pathway for their drug. The way that happens is something called accelerated approval, where you get conditional approval for a drug without the traditional 2 phase 3 positive trials, with like a condition, like an IOU to the FDA saying, I owe you more data, and I will give you more data showing that it's safe and effective in a larger group of people after it's been approved. And that is, I think, something that is actively being discussed between the company and the FDA. So I think that because of all of this that's happening with COVID-19 and accelerated emergency use questions, I think that's on everybody's minds. And it certainly makes sense for the ALS community to do that, because the risk-benefit ratio is very different for ALS than it is for example, a cholesterol medicine. People are willing to try things early, even if it's slightly riskier. And I think the FDA is starting to understand that. I hope. I think we're all working toward that.
So I have a message from somebody, but I can't seem to get to this. Richard I have an email, but unfortunately I apologize, Richard, because I can't get to this email. And I don't know if yep. Oh, okay. Never mind. So Dustin has a question. Why do they limit participation in the studies to only early onset, excluding people who have had ALS for a longer period of time? Mm-hmm.
That's a really good question. So I think that if we could have our way, we would love to include everybody in trials. From a scientific perspective, answering a question about whether a drug works or not. If you have somebody who's had the disease for more than 3 years, for example, that's the inclusion criteria for the platform trial, being less than 3 years in, the likelihood is that person is a slower progressor. And it's harder to answer a question when the changes are very slow. So the scientific community is trying to answer the question in a short period of time for everybody. And the best way to do that is to see if there's an effect on people who are moving quickly. I don't think I made that very clear. Having a limitation in terms of from symptom onset to the time they participate in a trial is trying to recruit people whose disease is changing faster. And that is a way of answering the questions faster so that the drug can be tested faster, so that it can be approved for everybody faster. If we had a lot of very slow changing people in the trials, like you saw in the tofersen group, the tofersen study, where there were only the non-fast progressors, there wasn't an effect. Actually, I could go back to that. Let's see. Let me see. Where did it go? Here. So this group of people were the people who are moving not that quickly in terms of their disease progression. And then this group of people were the people whose disease was moving fast. And even though we think that the drug is doing the same thing in both groups of people, for this group of people whose disease was not changing very much over 6 months, it would take years to answer that question. Whereas in the group of people whose disease were moving more quickly, we could answer the question in a few short months. And then the hope is that we could get the trial done faster so that we can get to the approval faster rather than doing a trial over several years to get an answer. That's the scientific reason for having a limitation for how quickly after onset people can participate in trials. And it's frustrating for the people who want to be part of research and who want to be part of the team and have access to trying something possibly, to not be able to. So I think that there's a lot of push for expanded access programs where people can try the drug, try an investigational drug under the guidance of a physician outside of a trial if they don't qualify because of how long they've had the disease. So that's the next frontier in trying to get more access for people who don't qualify for scientific reasons. A lot of the exclusion criteria for trials now, I know from the people who designed the platform trial, they really thought about it really hard because they wanted it to be as inclusive as possible for people to have access, while balancing being able to answer the scientific questions in a short enough period of time that they could test drugs fast. So there was a lot of back and forth about how to do it right for people, and this is where they landed after a lot of thinking. It's hard for the doctors who take care of people who want to be able to bring their patients into trials to not be able to bring everyone in, and that's why we're starting to think about expanded access on top of the trials.
Okay, we have a couple of questions that came into our Q&A. David has 2 platform trial questions. Can the regimen-specific screening be done virtually?
No, the regimen-specific screening can't, unfortunately. That has to be done in person because of some of the safety labs that have to be done, and I think some of the assessments that have to be done in person. I think the future of clinical trials, more things will be done virtually. I think that one thing this pandemic has taught us is that we should be more flexible, and we can be more flexible, and we should be, because it's a huge amount of work for people to come drive from Maine, where a lot of you are, to Boston for a trial, for example, to try to reduce the burden of these trials on people, especially for people with ALS. It's exhausting to do that kind of travel. So that's the future. Right now, the way it's designed, no, it can't be done virtually. If the pandemic becomes a bigger problem in the winter, and it becomes unsafe to see people, there are stipulations that allow us to do remote visits for some of the middle visits, but not the beginning and end.
Okay. And a second question about the platform is, how long after the last dose before data is submitted to FDA for approval?
That's a good question. I don't know the answer to that question. I know that Dr. Sokovich is very forward-focused and very efficiency-focused, so it will be done as quickly, faster than before, because that's her standard operating strategy, is be as efficient as possible and more. I think it depends on how quickly the data can be cleaned. So the cleaning process is when all the data is collected from people in trials. So you go into a trial, and the doctors and coordinators and nurses do all sorts of tests, and then they put the data into these databases. There are independent monitors that have to come in and check each and every single piece of data to make sure it's accurate, check it against everybody's medical records, and then check to make sure it's entered correctly. And so it's a huge amount of data cleaning work that happens. And then after that, the data managers have to make sure the data is clean from a database perspective. And then the statisticians have to look at it, and then it has to go to there's a ton of back end stuff that I didn't know about until I became part of this group. But it takes a number of steps, and then the company has to take over it. So I think it depends on all of those factors combined. And it always takes longer than you'd like. But I think that because of who's involved in these trials, they understand that there's a lot of urgency for all of these things. And it makes sense financially for the companies to move as quickly as they can, too. So that's part of the reason why companies are interested in working with the platform trials, that it just speeds things up, at least the trial part. And then the data part is a different group of people.
Okay, and that pretty much answered David's second question about explaining post-study scrubbing. Excuse me, let me just see if there are any additional questions. That seems to be it. As far as questions do that one. Okay. And you can wait, another couple of minutes here, but that seems to be it for the questions. On that, I'll give a couple of minutes to see if anybody has any last minute questions. And I want to take this opportunity to thank you so much, Dr. Chu, for taking time out of your, what I know must be a very busy schedule, to join us today and bring this information to our community. So thank you very much.
Thanks so much, everyone, for having me. This was lovely. Hope you have a nice weekend, and hope to see you in our trials if that's something that you'd be interested in doing.
Okay. So I think that looks like it's it. We don't have any other additional questions. I don't see any in the Q&A either. So just to remind everybody that immediately after the session, these are recorded. You can go back and listen to them. You can get back into this platform and watch the presentations again. So what we'll do now is we will stop the broadcast. Oh, thank you, Donna. That was great. We will stop the broadcast, and there will be a lag time. And the next thing up is the gamifications. And Karen is going to talk to everybody about how to get points and win fabulous prizes. And Dustin says, thank you for a nice, clear presentation. Great info from Richard. So again, when you see the stage, you'll see a blank page, and then we'll see you at the next next session. Remember, there's a little drop-down menu that will say "Join Next Session" if you haven't clicked on the next session. So again, thank you, Dr. Chu, and we'll see everybody later. Bye-bye.